So we're taking 10 mils of water from close to the surface of the tank and putting that into a small glass excitainer
septum.
I can come to you.
So we're going to take out 0.2 milliliters of air.
Taking out air so we can maintain the pressure.
And we're going to put in 0.2 milliliters of acetylene.
We're putting in 2 mils of acetylene gas. And this one is now
started,
ready to go. And in a moment we'll put this back in the tank and we'll let it go for
4 hours.
4 hours, and then we'll come back and add zinc chloride to stop the reaction.
Yep.
Going around to these different tanks, we are bringing a tote with all of the containers and equipment that we need. We've got our acetylene gas here. We've got our excitaners here. We've got DI water to rinse syringes that we use in between taking samples. And then we've got these other syringes, the ones that we use to remove some of the head space out of the excitainer and replace it with the acetylene. So these are like mobile field acetylene reduction assay gear tub. And we're going around to these different tanks that we filled up next to their respective ponds. Our acetylene reduction assays are underway in those tubes that are floating at the surface. And now we are going to take a water sample so that we can get nutrient analysis done, as well as chlorophyll analysis, and we'll collect a sample for phytoplankton coming up right here. So Kaylee's just getting a filter going. It's going to go onto a syringe. And she's going to pull, what, 50 mils?
Yeah, it's easy.
Whoa.
Yeah, tree.
We chose this tank because it was next to a tree. Getting it right to 50. Now we've got the water filtering through the syringe and into a falcon tube. And then we'll hang on to that sample until we get back to the lab and can process it. Okay, the wind is picking up a little bit here, which is fine. It's actually a decent day. We're gonna get some more sun later in the afternoon, I think, which will be nice. But we're still at tank 1 here. We just finished taking the nutrient sample, and basically what we did, we filtered 50 mils of water through this filter and collected that water in a vial. And this is what we're gonna get nutrients from. Then I filtered even more water through. I ended up filtering a total of 200 mils through this, and now we're going to take what's collected on this filter and we're going to put it into one of these smaller vials or tubes, and then that's what we're going to use to do the chlorophyll analysis. Okay, so that is the filter from the first tank. 200 mils of water went through there, and you can see the color a little bit on there. So this one did have it's going to have some chlorophyll in the signal in the analysis that we do. But I'm going to just fold this up into a little taco and then put it into the tube, and then we'll actually cover it in tin foil so that it no longer reacts with the light. Okay, tank 1 again. This is the phytoplankton sample that we're taking. So we're going to fill this all the way up, and then we will concentrate some of it to look at through the flow cam. At tank 1, what I'm seeing is some larval insect, maybe Coroborus. I saw another swimming insect in there, and it is kind of murky and like kind of reddish in color. So this will be an interesting one to sample. Okay, this is tank 2. Again, ARA, acetylene reduction assay vials are at the top. And this tank just happens to look really green, so I'm hoping we'll see some phytoplankton in there. There's not much in the way of zooplankton or any kind of predators or anything. So this seems to maybe be a tank with one trophic level. There are some more tanks down that way from the ponds. So tank 3 is right here. I've taken some photos of that. 4, 5, 6 in the back. And I know that several of these have zooplankton in them, and so these tanks tend to look a little bit less murky, and you can see the bottom in them. Like in tank 2, you can't even see the bottom. I also know that over there, tank 5 has some snails in it, and I know for sure that some of these have some fish, and I'll report those numbers based on what we saw when we stock these to you soon. Okay, this is tank 5. Can see all the way to the bottom, and you can see right there, see if I can point, that's a snail. There's a couple snails in this one. There's 2 more on the bottom there. Can I zoom in a little bit? There, you can see those snails crawling around, hoovering up the bottom. I'm not sure exactly what they're eating, but I'm guessing those snails are kind of like detritivores and eat the benthic algae that grows on the sides and bottom of the tank. You can see there's really nothing growing at all on the sides of this tank. Could be because of the snails. So tank 5, and then tank 6 we'll take a look at in a moment. Tank 6. No snails in there, but I'm pretty sure that there were some... oh, you can kind of see my reflection in there. Hello. Oh yeah, there's a snail down there, and I believe there are some zooplankton in here. In fact, I can kind of see some. It's probably impossible to see through here, but I do see some crawling around on the sides of the tank. You can probably just barely make those out. Those are most likely Daphnia, some Cladoceran. Alright, we got a little rain. Okay, so this is our zooplankton toe. It is 64 microns, and we are going to, for each of the tanks, we're going to take 2 toes from the bottom of the tank to the top. And the idea, which I think was decided upon by the group, that we would take 2 pools kind of in different parts of the tank. And so what I've decided to do here is to take the first one right in the middle of the tank, and I'll give that a quick second just to kind of hang out, and maybe if I scared any plankters, they'll come back. And then I'll take the second one on the edge of the tank to try to get, sometimes things like to hang out on the edge. So we'll go ahead and take this first one, let the water come out. That one didn't have too much stuff in it, so it's draining nice and fast, which is good. And then I'm literally just going to send it right back down, and we'll take the second toe on the edge. That. Think about what it's done. There we go. That's it. 2 toes collected into a single cod end, and that's our zooplankton sample. Okay, here is the plankton toe from tank 4, tank number 4. and you can totally see there's zooplankton there. There's a damselfly nymph, and lots of Daphnia, and maybe some other stuff. We'll have to look under the scope. But for sure, zooplankton. We have struck gold. Zooplankton tow. Okay, here's one of the samples in a jar that we have labeled, and it's gonna go into the lab, and we're actually gonna let these hang out for a little bit while we prepare the microscope area. We've decided that these plankters are a bit too big to go through the flow cam, so we are going to take photos of them under a dissecting scope and let you guys figure out their abundance and diversity from those photographs. I'm not sure how long these little buddies will last. They are basically larval or post-larval fish, I believe. You should be able to see them sort of twitching around in there. You can see my thumb for scale, they're quite small. But I believe these are the same as the sticklebacks that live in the experimental ponds and are often the focus of the experiments that they do here. But it's kind of neat to see them here coming out of our zooplankton toes. So I'll try to get a close-up look. Hopefully they'll survive long enough for us to look at them while they're alive under the scope. Okay, so here is the aquatic insect that I pulled out of tank 8 yesterday. We appear to be looking at the top side of this insect. So there's its head. Right there you can see its big eyes. It's got those big long legs, but interestingly, what you're seeing as the top of the organism is pretty light in color. And so I'm thinking that means that this is a backswimmer, because these insects have counter shading. So they have one side that's dark, so for things looking at them from above, they can't see them, and they're light on the bottom, so things looking from underneath can't see them, or at least not see them as well. And so what I'm seeing here is that the counter shading suggests that this thing should be swimming upside down most of the time, which would make it a backswimmer. The whole organism there from the side view, you can see it seems to have some kind of not sure, maybe a grasping claw of some kind. So it's a bit shaky. You can see that there. Maybe you can see that that's part of its feeding appendages, I think. Anyway, I'll try to get another shot. Okay, here's that insect again. I'm still thinking this is a backswimmer, but not totally sure. I've got it now so that its belly side is facing up towards me, up towards the light. And it does appear to be a bit darker in color on that side, which is the big thing that I thought made it a backswimmer. You can see it's really sort of stuck in the surface tension. These things do tend to carry a bubble of water on their bodies. Where'd he go? Okay, that it just poked its abdomen up above the water surface. So I think in nature it would actually do this, and then it would swim back down underneath the water and sort of has a lot of sort of hairs and things on the side of its body, can trap basically a little suit of air around its whole body. There, and it'll then swim underwater and do its thing. This is tank 4. Whatever that is not alive, but it was either a mayfly or a damselfly, and hopefully you guys can take a look at that and figure out what it was. Lots of Daphnia in this tank, like the other ones I've just been looking at. It almost looks like a Daphnia with another Daphnia on its face. I don't know what the deal is with that. You can see them flitting around. Okay, this is, appears to be, could be a harpacticoid copepod. I was thinking it could be an ostracod, but it's definitely some very small crustacean. And that looks like a mite of some kind. Oh! It keeps running away. Oh, I'm never gonna find it again. Anyway, that was a mite. I'll make a note of it from tank 4. Oh. There's our friend. Insect larva. Definitely seems to be predatory, although I haven't seen it strike at anything. I think this is what Corroborus is. You can see this. It's got a pretty gnarly hook on its head. It's kind of hard to see. Some of the other images will hopefully make it more clear. But it's likely that this thing is probably pretty limited in the size of Daphnia it can eat. So it probably can only really take the small ones for when the Corroborus is this big. It's likely a lot of the Daphnia you see in this image are too big for that thing to eat.